Hello, BIO178. In the previous video, we went through the basic workflow and analysis for genomics, and in this video, we'll go through the basic workflow and analysis for transcriptomic studies. So first, let's think about what type of information the transcriptome can give us. The transcriptome, again, is all of the expressed genes in an organism. So the genome is all of the DNA. The transcriptome is all of the genes that are actively being expressed at a certain point in time. So first I want you to think about, maybe you can look at this image here on the right, think about what type of information the transcriptome can give us. What do you think? So if we think about the different types of information, for the genome, as we've talked about previously, can give you information about the DNA sequence, about SNPs, for instance, right? The transcriptome can also give us that. So the transcriptome can also give us information about nucleotide variation. But what's unique about the transcriptome that it can give us that the genome cannot, it can also give us information about gene expression. So remember I said the transcriptome is all of the genes that are actively being expressed. When you do a transcriptome high throughput sequencing study, you can also get information about the level of expression of these different genes. So in this image here on the right, this is showing the process of going from your DNA here to your RNA transcript. That's then translated, your mRNA is translated by the ribosomes. So when you're doing a transcriptome study, by sequencing this RNA, you can work backwards and get information about nucleotide variation that was on that original template DNA strand. But by looking at also how many copies of this mRNA you have, which is what you can get from a transcriptome study, it can also give you information about the level of gene expression. So let's walk through now the basic workflow and analysis for transcriptome studies, and a lot of this is similar to the genome, so I encourage you to think about how it's similar and how it's different, and I'll emphasize here the differences between transcriptome and genome workflow. So the first step is we want to extract our what's the word I'm looking for? Nucleic acid, that's it, nucleic acid. Before we were extracting DNA, now we're extracting RNA. So we're still taking a piece of tissue from the organism, for instance, but now it's really important for us to think about what type of tissue we're taking and when we're taking that sample. And that's important for a couple of reasons. So gene expression, if that's what we're interested in, it varies a lot across different tissues. So even in the snail, if you took some of its foot tissue versus underneath the shell, somewhere here there's a gill. What else do they have? They have their gonads. So depending on what type of tissue you take, you're going to get different types of gene expression. So it's important to think about what type of tissue you're taking and when you're taking it. So for instance, when I talked previously about the genome dd-rad study I've done with my snails, quote unquote my snails, what I did was basically take a bunch of snails, put them in ethanol to kill them, and then I just took a piece of their foot tissue and extracted DNA. If you're extracting DNA, that DNA sequence is going to stay the same no matter what. Doesn't matter if they're male or female snails, doesn't matter if they're alive. But when you're taking samples for transcriptome studies, there it's really important to think about, okay, how am I going to get this tissue? When am I going to get it? So for that study, what I did is actually expose the snails to high temperatures, to heat stress, and then at that point, I froze them in liquid nitrogen and then extracted RNA from the entire organism after I took the shell off. So it was important for me in that case when I'm collecting RNA to make sure that I'm sampling them at a time point where I'm specifically interested in knowing what their levels of gene expression are. So for this type of transcriptome study, you're extracting RNA specifically from a tissue type or a whole organism that's been specifically chosen for your purpose. When you're doing the actual extraction from the tissue, often you do this fun liquid nitrogen grinding process where you kind of look like a witch with a cauldron, all this liquid nitrogen coming up. And because RNA is very unstable, you don't want to keep it as RNA. So what you do is actually convert it to cDNA or complementary DNA. So that's what's shown here. You're converting to cDNA, and that's what you actually work with for subsequent steps because the cDNA is more stable than RNA. So just like in the previous genomics video, you first want to fragment that cDNA and then create a cDNA library by adding indices. Remember, indices are those unique barcodes that allow you to demultiplex later, and the adapters are what allow the sample, the library sample, to actually bind to that flow cell. You then sequence that cDNA using the same types of Illumina machines, and then reassemble those short reads you get back, which are just like before, between 50 and 250 base pairs roughly, and you're reassembling them into the original mRNA sequence by still identifying those same regions of overlap. Next, you then, just like we did before, want to determine the function of those reassembled sequences, those genes that you reassembled. Here, it's going to be a little bit different because we started with RNA, right? So we know that every sample we have is from some gene that was being expressed. So here, we're expecting every one of the regions we've reassembled to have some kind of gene function because we're only looking at those expressed regions. Remember before with the genome, we were getting data from both the coding and non-coding regions. But now we know everything in our data set is from an expressed region. So to summarize, steps 5 and 6, where you're assembling and annotating, you start with your raw RNA-seq reads, which are those short 50 to 250 base pairs. That depends on the particular type of sequencing you're doing. You're then reassembling those short reads, identifying the regions of overlap to reconstruct this full-length gene sequence. And again, we're using that term de novo. So if you are assembling these genes from scratch where there was no genetic reference already available, for instance, this is what I had to do with the intertidal snails when I did a transcriptome study because there weren't any genomic references available, so I assembled all of these genes in my transcriptome study from scratch. So we're reassembling those different genes, and then we're doing that annotation step. Again, in this scenario, we have differently colored genes. So we have a blue gene that when you annotate that, compare it to a database, it might indicate it's a heat shock protein. A green gene might indicate it's a stress granule gene, and a red gene here. And remember, previously with the genome, we indicated this might be an intronic region where we don't actually know what the function is. But again, here, because we are specifically looking at the transcriptome at an RNA starting material, we know that these should all be actual expressed genes. And just like with the genome we talked about previously, you can investigate nucleotide variation from this sequence data you're getting back in the transcriptome. However, note here that those SNPs we're identifying, if we are identifying SNPs, are always in coding regions. If you're looking at the genome, those SNPs can be in coding or non-coding regions. Here with the transcriptome, it's only coding regions. And the unique thing here that we can do with the transcriptome data is get gene expression count. So we're able to look at the levels of gene expression. And we do that by doing something called mapping reads, which I'm illustrating here in this graphic. So say you have two different samples. One of your samples you took from a group of animals that was exposed to cool conditions. So say they had not experienced any warm temperatures. Your second group was a group that was exposed to warm temperatures, and you sampled them after being exposed to that. So that's often called heat stress. So you have one before heat stress sample, or your control sample, and one after heat stress sample. So here in this picture, we have that full-length sequence of, say, a heat shock protein that we were able to determine by reassembling those short reads. That's the de novo transcriptome assembly we talked about previously. And then what you can do is use those same reads, the short reads that you use to reassemble that full-length gene sequence, And you can see how many copies of that gene you actually have in your sequencing data. So basically, the number of reads you get in your sequencing data that correspond to one particular gene region can be used as a proxy to determine how much that gene was expressed. So if you see in your transcriptome data, you have a bunch of reads that came back that map or match one particular gene region, that means that particular gene was expressed really highly at the time point that you took your sample from your organism. So here in this example, say under our control conditions before heat stress, there are 4 reads that map to this heat shock protein. That would give us an expression count of 4. Say after heat stress, we now have 8 reads that map to that same heat shock protein. That would give us an expression count of 8. And we would say that this particular gene was upregulated or turned on following heat stress. So in response to heat stress, this particular gene was expressed at a higher level compared to what it was previously. And after mapping genes, what you can do is determine genes that are significantly differentially expressed between 2 different treatment groups that you're comparing. So this is a figure from that Fraser paper that I referenced earlier, looking at the bobcats and their response, their gene expression response in one group of bobcats that had been exposed to these anticoagulant poisons and one group that had not. This type of graph here on the left is called a volcano plot. So it kind of looks like a volcano erupting. I think that's why they're called that. But the way to interpret these is each one of these dots is a different gene. And what we're looking at here in terms of where those dots fall, if those dots are above this dotted line, so basically everything in this region here that's in tan, all of those genes have significantly differential expression between the 2 comparison groups. So they're either much more highly expressed in one group than the other, or vice versa. And so what you can do is identify genes that have significantly different expression levels. You can do that, look for significance in packages in R, for instance, and then you can pull out those genes and see what those genes are. So in this case, if we look specifically at some of those genes and their functions, again, we identify those functions by mapping them to those known databases. We can see that a lot of these genes, all these CD3 genes, they all are related to immune functions. And so in this graph on the right, we're just pulling out individual genes and comparing the groups. So these are individuals that were exposed to that anticoagulant rodent poison versus these individuals that were not exposed or negative. And we can see that for these 3 genes, the bobcats that had been exposed to the anticoagulant poison had higher expression of these immune genes. So that was one of the differences they found in this data set, is that the immune gene function for a lot of these bobcats that had been exposed to the poison, those immune genes were upregulated, so they had differential expression for those particular functions.